So I'm really excited to have Dr. Joe Raffaele. We actually only met a week ago at the Longevity Roundtable Conference. And I was lucky enough to hear you speak at this incredible conference. And up until then, we hadn't been introduced. And I was really quite blown away by, well, your brilliant intelligence, goes without saying, but your depth of understanding of this world of longevity and all the things that need to come together to really find answers. So I'm going to hand it over to you. And if you could just introduce yourself, the work you do, the platform you built, what is it that makes you work differently? And then maybe I can ask you a few questions after that.
Sure. Yes, so I have been a longevity medicine physician for 25 years. We used to call ourselves anti-aging physicians, age management, but I think longevity is the proper term at this point. And I got interested in it. I'm an internist by training. I got interested in it because I went to a conference in 2000 or so, and finally got to meet Bob Butler, who was the late founder of the National Institutes of Aging. And he said, "You're doing great stuff, this new field, focusing on aging, but what do you do to tell your patients you're actually aging better?" And I said, "Oh, good question." and he said, "Yeah, you seem like a pretty smart guy. Are you looking at any biomarkers of aging?" And I hadn't been at that point because patients were doing well. We're doing their hormones, their supplements, their exercise. And I said, "Okay, let's look at the literature." And I spent the next 20 years looking at the literature, and there are a whole bunch of biomarkers out there that now we are all talking about. But we started with the more non-blood ones, the arterial stiffness and pulmonary function. But the fundamental concept is if you're in a pre-disease state trying to alter the aging process, just like with blood pressure, you have to have a metric. And that metric is, multiple different ones for different parts of your organ systems, your lungs, and now we're looking at genetics, epigenetics, glycobiology, all these things. And so what I then did in my practice to track all that is I developed a software platform for all that data to go into it because, as the typical EHR doesn't really handle that well. And now that's called PhysioAge Health Analytics. And with that platform, I can track longitudinally in a very simple way. I can just click and know what's happening in this marker compared to this marker. I have practice level analytics where I can see how, what's the worst hemoglobin A1C, the best one in my practice, what's the average one. There's called our data clouds for our over 600 parameters in there. And I mean, that's really how you practice evidence-based N of 1 longitudinal longevity medicine. And that's what I do. A little bit of focus on telomere biology as well, which is the sort of only malleable part of the genome. I kind of think of it as the telotype rather than the genotype. But that's what I do. And then I met you and listened to your brilliant talk. And if I can just go on and on, but let me just say that I'd gotten my genome sequenced in, I think, 2015 or something when Illumina was doing it, like know your genome thing. And then I was really excited to get the report back. Then I looked at the report and I'm like, this is not telling me anything useful. I mean, I know I didn't have any of the mitochondrial diseases, so that's great. Little propensity to AFib, but other than that, just getting information from it wasn't useful. And even ever since then, I was looking at all the genetics companies. It wasn't actionable, or they were saying it was over actionable, which, making claims about what a SNP does that perhaps isn't really, because my job is really to get into the deep science and the evidence, translate it for my patients. And I think, I hadn't been using genetics until now. I think I'm going to start doing it.
Yeah. So there's nothing more interesting than meeting someone at a conference which was data heavy, right? So many clinics doing whole genome sequencing, lots and lots of genetic information. And the one doctor who turns around and says, so everyone's doing whole genome sequencing, and Dr. Joe turns around and goes, I'm not doing genetics at all. And I'm like, you're probably the only person in this whole conference not doing genetics. And I was like, why? And you're like, well, I've never met a company who I felt was using genetics in a clinically useful or actionable way. And I think that's when our conversation started. And I think that's where you and I really agreed, because what I was hearing too much of is genetic for data's sake and not genetic for the insight. And it sounds like you've built your whole platform on how do we take data and how do we gain kind of clinical information that I can actually action. And that was where I think we're coming from the same place. And it was really a kind of a delight to hear you say, well, actually, I've never met a genetic testing company who made sense and who's focused on what is the clinical value of this information rather than kind of just collecting more and more data on our patient. And so maybe, so thank you for that. And we're very excited and look forward to working with you. But maybe as we've just come out of this conference, of all the things that we learned last weekend, aside from finding a fantastic genetic company, what do you think the one kind of take home that you can say that you learned something new there that would be exciting for you to kind of take forward to your patients?
I would say first that I do some genetic testing, genotyping for the very well characterized ones like ApoE and hemochromatosis, et cetera. But beyond those, I wasn't doing much because those are very well validated. You know what to do with them. But I think learning from you was one of the things was how those very well known ones can actually be modified by lesser known ones.
Yeah.
Like, I mean, this is not a plug for you, but this is one of the big take homes I took from that meeting, which was my one ApoE4, thank God not two, is modifiable by alpha klotho, by Tom40, and I have 25% of my patients have one of the ApoE4s because I've got a pretty big practice, and that's population statistics. And so now I can go to them. And I was always telling them that it still is, with the Bredesen protocol and other things, lifestyle changes, that it's not, a sentence for Alzheimer's disease, but learning that there actually are some really good markers that can tell them. I mean, they could be a little bit worse with some of them too, but overall that there's another way to get more granular about it, I think was one of the big takeaways. The other one, the big one, which I've actually been preaching myself, but it was great to hear that it's been echoed, is that there's no single biomarker of aging. And there's no single genetic test either that's going to, except Huntington's probably, that will characterize your biological age. It's multiple biomarkers, multiple systems. They can be 25 years apart. That doesn't mean they're wrong. It just means that one system is aging better or had more genetic inheritance versus the other system. And you got to treat each one with your therapies focused on that system.
Yeah. And I think that's just it. So, the same way at 3x4, we think about pathway-based genomics, like let's look at the pathway, let's look at all the genes that influence it, let's group them together. I think what you're saying is it's absolutely true of genetics, it's true of biomarkers, and it's true of everything else. And hopefully we'll find a way for the future not to get stuck in the kind of noise of data and really find a way to make sense of all this data and put it together and be better clinicians. So thank you very much, Joe. Thank you for joining us here today. Really appreciate it. And it's great seeing you again and looking forward to seeing you many more times.
Well, thank you. I just want to say, and I'm really excited to get my results back
Yes.
and get started with my patients. So thanks for creating 3x4 Genetics.